So, since the middle of the last century, 2 billion hectares of land has been degraded globally. 2 billion hectares of land is an area about the size of Russia, so it's pretty big. The Bond Challenge aims to restore 350 million hectares by 2030. Not quite 2 billion hectares, but a substantial amount. There will be increasing interest in how we do restoration, how we source seed for restoration, and also how we monitor the outcomes of that restoration. So that's pretty much the context of my talk and what we should be doing here in South Australia about restoring our native vegetation. So the key challenges with vegetation and restoration activities are around habitat fragmentation and climate change. So let's keep these in mind. And the three issues that I'm going to talk about in some detail is, can we source seed from isolated trees in the landscape? It might seem like a small question, but that's where many seed is sourced from for restoration projects. Should we be looking at local provenancing, or have we moved beyond local provenancing? And how can we improve monitoring of restoration outcomes? So, by way of context, I think you may have seen this already. Greg Guerin did a piece of work to analyse biological survey and herbarium records for the distribution of plant species here in South Australia. 6 biodiversity hotspots have been identified, and they're threatened by habitat fragmentation, invasive species, and a cocktail including climate change. And we can see on the map the large amount of removal of habitat, particularly in the southern part of the state. And this is the key area that most of the restoration activity is currently ongoing within. Now, I'll just draw your attention to the blue lines that are going across there, indicate areas of maximum predicted turnover in community composition under future climate. So it kind of gives an indication of the sensitivity of the communities under predicted future climatic change. And rather satisfyingly, it's around Goyder's line. So anyway, so interesting context for restoration work here in South Australia. So why shouldn't we source seed from isolated trees? So this is a mahogany tree in Costa Rica. This is my friend Carlos, who's collecting seed. The lazy lad couldn't be bothered to go into a forest to get a good sample of seed. He drove his Hilux underneath a tree that was isolated in a farm context where it's very easy to collect seed from. And this is a common source of seed for these types of projects. Why shouldn't we source from these? Because we know that isolation of habitat causes reduced genetic diversity, increases genetic differentiation, and increases inbreeding and inbreeding depression. This is the theory of population genetics. When you reduce gene flow between individuals and between populations, you get these type of effects. And what we think is this leads to an extinction vortex where small population size increases inbreeding, decreases genetic diversity, leads to smaller population size, and so on until you go down the plug hole of life. But it's not quite that simple. I won't show you the slides, but what we did is looked at some of the variation in natural populations, and we find that the trees are actually a little bit robust to these kind of impacts because they have long generation times, and because their gene flow mechanisms are actually pretty robust to habitat changes. And so we don't see massive decreases in genetic diversity or genetic differentiation, but we do see real inbreeding effects within the seeds of isolated trees. This is a meta-analysis that was done by Martin Breed, his postdoc in my group, and this is done as part of his PhD project. This is an analysis of global studies looking and comparing mating system parameters in trees and this is for seed that was sampled from either a continuous forest or a fragmented forest, or from isolated trees. And generally what we see is for seeds that are sourced from an isolated tree or a fragmented context, we see a big increase in inbreeding. And we also see a loss of pollen diversity. Both of those have significant impacts on the growth performance of seedlings. So seedlings that are sourced from isolated trees have inbreeding, they have less pollen diversity, and they do poorer. So we should be avoiding isolated trees for a seed source for restoration and focusing on seeds coming from a more continuous context. Right, next one. Climate resilient provenancing. Again, back to Costa Rica. These two trees are playing soccer, and you see it's quite regularly. And one of them saying to the other, I always play better at home. But this isn't always true, and we could talk to the English rugby team of the last Rugby World Cup, who clearly didn't play better at home. So sporting analogies are good, but I do like it when sporting analogies don't work. So why wouldn't a local provenance be best? If you can imagine, this is a kind of fitness curve. So you've got a climate gradient, you've got a particular climatic type, and you've got maximum fitness in the middle of that climate gradient. So your tree species does best in this area here. So if that holds, that's good. You do want to source then your seed from this area. But say you source your seed from that central bit, the best possible fitness, and then climate change comes along and it moves the climate so that particular source location is no longer as fit as it used to be. What you're hoping is that actually, as you're sourcing seed, the populations are going to keep track in terms of adaptation with the shifting climatic change that we've seen. And we've seen a lot of climate change over the last 50 years. Let us be clear that in the last 100 years, we've already gone through 1 degree of temperature change. In some areas, we've witnessed 15 to 20% rainfall decreases. So what does this mean in terms of restoring South Australia's vegetation? This is some work done by Nick Gelly, a PhD student, and just looking at the different climate envelopes along the York Peninsula here. These are different provenance locations, the different squares there. And we can see, moving to 2100, we can actually see that the climate at the northern part of the York Peninsula moves south. So what was a warmer, drier location at the northern part of the York Peninsula then becomes that climate in the southern part of York Peninsula. And interestingly, what we found, and Nick is still analysing these data and going back and measuring this provenance trial, but we found that local provenance does not do overwhelmingly well in this particular location. So, Point York trial for Eucalyptus oleosa, we can say that local provenance does best. Does this pointer work? There we go. So, plant height here, this circled one is the local provenance, and it does better than these three. But look at this one here, Eucalyptus porosa. The local provenance here does worse than provenances from each of these other locations here. So we found some disturbing evidence that shows that local provenance doesn't do very well at all. So we were interested in this, so Nick and Martin organised a workshop and brought together some of the key people that have been involved in provenance trials around Australia. So there's a very good list of plant geneticists there, and they pooled all of their provenance data, 634 provenances, 39 different species, almost 100 unique trials. And this is when the provenances were planted, year monitored. Sorry, this is the year that the provenance trials were monitored. And then this is a relative performance where local provenance does best, 1 here, or really bad, does 0 here. So some of these provenance trials were quite old, and what we see is that the local provenance does well, or best, when they're first established. But actually, these dates here are actually 30 years since the provenance trials were established. And what we're seeing is that over time, the local provenance doesn't do as well as other provenances in these provenance trials. And that's consistent with our understanding that the climate is shifting over time. So that holds. But the really disturbing finding was this finding here. And there have been a number of new provenance trials established, largely by Nick and Martin, in the last 5 years. And what we see is we would expect the local provenance to do best here. We would expect a resetting, but we're not seeing that. We're seeing that local provenance really doesn't do very well at all across many, many trials. So local provenance did best in 16 out of 73 cases. We're seeing a decline in local provenance performance over time, but more disturbingly, recently, there's a breakdown, apparent breakdown of local adaptation. That's a very significant finding. So, we shouldn't go near provenance, local provenance. No, okay, so let's steady. Okay, steady. So, local provenance, there's still some element of adaptation, but we need to consider mixing provenances from other sources, particularly from warmer, drier locations. Still use local provenance as the basis because there's likely to be a range of local adaptations there that still persist, but having the opportunity to bring provenances from areas which are likely to be warmer and drier in the future are likely to give those plantings adaptations that they will require for future success. How am I doing for time?
3 minutes.
3 minutes, excellent. And we still have a long way to go. This is released in April. Local is still called best. Local is not best necessarily. And so Society for Ecological Restoration is still putting out information that local is best. So we need to get on and publish those results, Martin and Nick, and I'll leave that with you. Good. How do we quickly and easily assess restoration outcomes? So, a lot of restoration is assessed by stems per hectare. You get some money from federal government, you stick some trees in the ground, and then you go, okay, number of stems per hectare, that's our measure. That's a measure of an input. Or you might have the Society for Ecological Restoration. It kind of has an idealised goal, a motherhood statement around return of ecosystem function. What is ecosystem function? We don't really know, but it sounds good and we should have it in spades. I don't mean to be too mean to the Society for Ecological Restoration because they're doing good work, but it's a useful framework. Yeah. Now, what we do need is smart, measurable KPIs, so key performance indicators of restoration. We're not going to go out and measure everything about a restoration planting, but we do need some good indicators of ecosystem function. So, again, this is work done by Nick and Martin. Maybe you should have given the talk, I'm sorry. So this is looking at a restoration sequence. So we had this is cleared sites through to remnant vegetation, and this is years ago since restoration activity took place. It's a replicated design across the landscape, and we looked at the soils and did DNA metabarcoding of soils at this site. And we found some quite interesting findings. So this is principal component analysis, so you can pull apart the variation in the data set. Cleared sites over here in red, and we see the younger sites to the older sites going through to remnant vegetation over here. So that's my time, is it?
1 minute.
Oh, great. Increasing time since restoration. So what we also see is soil characteristics, increasing soil carbon, decreasing nitrogen and salinity. But what we do see is after 8 years of restoration, the soil microbiota has moved significantly towards remnant sites. So very promising. 8 years of restoration. And you also see changing communities that are linked with key functional return at the sites. Now, there is now a national database of soil microbiome information which can be used to compare restoration sites against a baseline that occurs out there. So our recommendation is that DNA barcoding is now ready to be used as a monitoring tool in restoration.
Excellent.
Thank you.